Welcome everyone to the Hedimed online stakeholder event. My name is Jutta Laiho and I'm a bioscientist at Tampere University, Finland, and I'm the scientific manager of the project. So I will be chairing today's event with the help of our scientific management team. And I also want to introduce now our project PI, the principal coordinator, Heikki Hyöty, professor of virology at Tampere University, Finland, and maybe Heikki wants to say welcome words as well.
Yes, thank you, Jutta. I also would like to welcome you all here. So this is a large EU study, very multidisciplinary. We have a lot of, I think, new openings and new innovations in the project, new technologies. So please be active, ask questions, and you can also always contact us so we can then reply you after the meeting by emails. Thank you, Jutta. So just go ahead.
So we have invited researchers, policymakers, and public and health experts today, and all the other stakeholders to this event. And we're very happy to see that so many of you are here. The aim of this event is to introduce you to Hedimed project and also to demonstrate the science and results that we have obtained during the past 5 years, and also showcase the innovations and technologies. By doing that, the aim is to foster meaningful dialogue with an external audience, meaning you guys. Let's go through the agenda of today. Now, after the opening session, we will briefly introduce you to the Hedimed, the exposome concept in general, and also we will have European Commission representative here to tell you about the secondary use of health data in exposome research. Then we will have, in the first session, we will dive in more deeply into the Hedimed research. So you will have we will have 4 presentations from Hedimed researchers about exposomic insights into immune-mediated disease risk factors. Then we'll have a short coffee break, and after the coffee break there will be another session which focuses on the innovative technologies and platforms for exposomic research. Then we will have a lunch break. Next session is the exposome research related impact on policy with 2 presentations. Then we will end the day on panel discussion. So we will have 6 people eager to discuss about how exposomic insight can shape the policies and public health. So we can actually now start to go to the first presentation of the day, which is the introduction of Hedimed and the exposome concept. So you get a bit of background why we are here today. So Heikki and myself will give this presentation. So let's start with the exposome concept. So what is exposome? It means the totality of exposures to which an individual is subjected from conception to death. So these include factors resulting from environmental agents, socioeconomic conditions, lifestyle, diet, and endogenous processes as well. Why should we study the exposome? Well, genome-wide association studies, or GWAS studies, have shown us that genetics account for only a small percentage of diseases. So there must be another causative thing there. So it's coming from the exposome, environmental factors, most likely. So by characterizing the exposome, it enables us to address the possible associations with the exposomic factors with health outcomes. And also, we know their significance alone or in combination with the genomic factors to the disease outcomes. And that was a small introduction to exposome. And then Hedimed stands for Human Exposomic Determinants of Immune-Mediated Diseases. The project is funded by the EU's Horizon 2020 program, and it started January 2020, and it's a 5-year project, 60 months, but we got 6 months no-cost extension to the project, so the project will run until the end of this June, so a little over a month still. The total funding was around 12 million euros. What is the rationale for the project? So first of all, immune-mediated diseases, or IMDs, they have been increasing rapidly in developing countries during the past decades. And this increase is so fast that it cannot be explained solely by genetics. So there must be an environmental input. So you can see here, if we look at the the urbanization, for example, starting from the 19th century, the urbanization has increased, and at the same time, the immune-mediated diseases incidence has increased. And at the same time, the rate of infectious diseases have gone down, and also so has the biodiversity. So there's something going on there. Second of all, there is no curative treatment for IMDs at the moment, and we need preventive treatment. So to have those, we need an understanding of the disease mechanisms, especially the ones that operate early in life, since this is the time when the immune system is developing rapidly. So Hedimed has many study hypotheses, but here is the main hypothesis right now. So we think that there are multiple effects of the exposome that affect the risk of immune-mediated diseases. So there are exposomic risk factors that are necessary or sufficient causes of IMDs, such as gluten intake in celiac disease. Then these risk factors affect the immune system or organs or cells that are damaged in IMDs. Then there are also exposomic protective factors that promote the regulation of immune system and also control the enhanced immune responses. And there are modifying factors that modulate the effect of risk and protective factors. So what is Hedimed about? In Hedimed, we want to identify and model the exposomic factors that are relevant for IMDs. And in Hedimed, we're especially focusing on 4 disease outcomes, namely type 1 diabetes, celiac disease, asthma, and IgE sensitization or allergy. So we want to find out whether we can identify exposomic risk factors or exposomic factors in general that play a role individually in these diseases. Oh, the exposome is huge. There are so many factors that one can study, but we need to focus on some factors. So here's a list of the key exposomic factors that we're studying or have been studying in Hedimed. So these include microbiome, infections, environmental biodiversity, toxins, dietary things, psychological stress, then more specifically extracellular vesicle content. So these are the small vesicles in our cells. And also host response to exposomic factors has been studied. The focus is on early life events, as mentioned. That is the time when the immune system is rapidly developing. And for this, we have had a beautiful selection of clinical cohorts available for Hedimed, spanning from pregnancy time to childhood or birth, early childhood, and until adolescence too. But we're focusing especially on the early years of life and pregnancy time. We also have 4 clinical trials included in the project. Here's a list of the specific aims of Hedimed. So there are a few of them, but I will go through them because they kind of give you an idea what we have done during the past 5 years. So first of all, we wanted to identify the disease-specific exposomic determinants of different IMDs, including the fetal and postnatal exposome. We also wanted to identify the exposomic determinants that are shared by these diseases. We wanted to characterize biological pathways and mechanisms that mediate the effect of the exposome. We wanted to identify factors that modulate individual susceptibility to exposomic influence, meaning like age and gender and so forth. We wanted to identify the interactions between different exposomic determinants in the development of IMDs. We wanted to develop new sensor technologies for the characterization of exposome and its role in IMDs. We wanted to develop methods for modeling the effect of the exposome. We wanted to develop prediction models to estimate the health economical savings generated by prevention of exposomic disease determinants. We wanted to build a toolbox that offering data, know-how, technologies, information, data analytical and modeling tools. And obviously we wanted to disseminate and implement the findings in collaboration with various stakeholders like we're doing today to facilitate the development of novel diagnostics, therapeutic and preventing approaches for IMDs. And lastly, we wanted to test if interventions targeting the exposome of young children can reduce the risk of IMDs. And this is why we have the clinical trials in Hedimed. So who are we? So this is the Hedimed scientific management team. Hedimed is coordinated from Tampere University, Finland. So Heikki and myself, we already said a few words, but in addition, we have Olli Laitinen, who is going to today later on moderate the panel discussion with me. And then we also have Leena Puustinen and Henna Numminen in the room here and helping with a variety of things. We have 22 partners in Hedimed coming from 12 different countries. So we have one partner from the US, but all the others are from Europe. We have the partners are mostly universities or research organizations, but we also have 3 small or medium-sized enterprises in the project. As mentioned, the clinical cohorts play a very important part in Hedimed, and they enable the studies that we have done. So this is a bit heavy table here, but this is to show you that we have a number of cohorts in this study. So they come from different countries, but mainly from the Nordic countries, Sweden, Denmark, Finland, and Norway. And we have, especially the prospective birth cohorts are important in Hedimed, and Heikki will say a few words about that in the next slide. But we have different types of cohort. There's a cohort size that available for Hedimed. We have not used all samples, but this is the size that we have available. You can see that many of the cohorts have a time point data or samples from the pregnancy time point, and many of them also have samples and data from birth until the young adulthood. So this is time in age range in years here. Then on the green here, this is just to show you quickly the sample and data available, meaning, for example, blood and stool samples, which were mostly used in our studies. Also, nasal, skin, and spirometry samples have been available in some of the cohorts, and then questionnaire data and other metrics are available. And to conclude, pretty much, so these cohorts cover all the endpoint diseases of Hedimed, so that's very important. But now I'm going to give Heikki the floor to talk this slide through.
Yeah, well, this may be the only slide I talk about, but I want to also emphasize the importance of the prospective birth cohort studies in this kind of project we are carrying out. So we talk about chronic diseases, which are usually start way before the symptoms appear. And there are biomarkers we can use to find out which individuals actually have a subclinical process, which is not causing any symptoms, but which eventually leads to the disease. And that's why the what we call prospective means that we follow children from birth. And that's very expensive. That's really a huge effort to collect this kind of cohorts and then following them for, let's say, until age of 15, for example. In many studies, we can find out that some of them develop the diseases we are studying, and then we can go back to the freezers and look at the samples we have collected from birth and analyze them for different exposomic variants and also the host response patterns. And we have interview data about the diet or other things which are important exposomic components. I think we are very privileged to be able to collect so many good cohorts together. So we call them cohort owners. So they are the cornerstone of the whole project, the high quality samples and prospective design. Maybe that's all I want to say about this one.
Then one slide about the practical work of Hedimed. So Hedimed was, we have in Hedimed 13 different work packages. We have focused on specific external exposome, general external exposome, and also internal exposome. And also we developed these new technologies, had organised and cell models, all that. Also ethics were packages because we have clinical cohorts and ethics is very important, obviously. I just want to highlight here the work package 12, which is European Human Exposome Network activities. So at the same time when Hedimed was funded, there were 8 other exposome projects funded under the same call. So together, us 9 exposome project, we formed the European Human Exposome Network. So it's a huge network. So if you want more information, please visit humanexposome.eu for more information. So all these projects started 2020 and they will end by the end of June. And I just very quickly want to show you some couple of last slides here. So in Hedimed, we have had around 50 different research projects. And so far we have published around 20 scientific articles, but most articles are still to come since we are in a very active phase now, just trying to finalize things. So the publications most of them come after the project ends. We will update the Hedimed web pages so you can find the information from there. Then I want to highlight on the web pages, you can also find blog posts that are in the core of Hedimed. These are very clear, so they are very in our opinion, interesting too. Why do we need pair cohorts? How satellite data can be used in health research? What is the nature's role in allergies? The eating exposome, what you eat, how does it relate to like type 1 diabetes, for example? Then there are blog posts about the technologies that have been developed in Hedimed, and from some of these you will hear presentations today as well. And also soon we will have online all the tools that have been developed in Hedimed, so there are quite a few of them. So just wanted to advertise this as well. So I end my presentation here.